Hello, everyone, and welcome to the latest installment of Scrubbing the Skies, the webinar series of the Institute for Carbon Removal Law and Policy. As always, I'm Will Burns, the host of the show, and I serve as the Institute's co-director. The Institute for Carbon Removal Law and Policy was established to provide a platform to scrutinize scientific, legal, political, and ethical questions associated with the emerging carbon dioxide removal sector. So let me initially frame up the focus of today's webinar. The National Oceanic and Atmospheric Administration's recent announcement of allocation of $24 million in funding for marine-based carbon dioxide removal accentuates the increasing focus on how the oceans, which already comprise the world's largest carbon sink, might be called upon to do more to combat one of the gravest long-term threats to oceans, among other areas, climate change. Perhaps the most prominent approach in this context is ocean alkalinity enhancement, an abiotic approach that aims to increase the ocean's carbon dioxide uptake capacity through the dispersal of pulverized minerals or dissolved alkali into the surface ocean. Now, while many believe that this approach could hold great promise in terms of enhancing uptake of atmospheric carbon dioxide into the world's oceans, serious questions still abound about the potential impact that such interventions could have on ocean ecosystems. And while this question has been largely underexplored to date, today's guests have just published a new piece in the journal Science Advances that seeks to enhance our understanding of how alkalinity enhancement interventions might impact oceanic species, with a focus on representatives of two important phytoplankton groups. And so let me introduce today's speakers. James Gately is the lead author on this piece and is a graduate student in the Department of Ecology, Evolution, and Marine Biology at the University of California at Santa Barbara. Deborah Iglesias Rodriguez is chair of the same program at Santa Barbara. James, Deborah, welcome to the show. So we'll begin now with a brief presentation by both James and Deborah to summarize the article, followed by a few direct questions by me, and then we'll open it up to the audience. So please populate the Q&A box with questions as the webinar ensues. And with that, I'll turn over the podium to James.
All right, thank you, Will, and thanks for having us here. I'm excited to share our work. Let me share the screen. Great. Yeah, so as Will mentioned, we're going to be discussing our results where we looked at representative species of Coccolithophore and diatom in their response to ocean alkalinity enhancement. So I'm not sure what everyone's background is here, so I thought I would give a brief introduction to ocean alkalinity enhancement. And as Will said, it involves the dispersal of pulverized mineral or alkali into the surface ocean. So there's two parts. Like broadly speaking, two parts to this. The part one, which occurs on land primarily, which involves the mining of minerals. Some proposed minerals are like limestone and their derivatives, or perhaps olivine. And other options include the production of an alkaline solution through electrochemical methods, maybe like sodium hydroxide. But part one would occur on land, the production side, and then that would be the alkali would then be dispersed into the ocean, either coastally or in the open ocean. And this could be done, through like a pipe running into the ocean, dispersing an alkaline solution, or perhaps the pulverized mineral being dispersed in the surface ocean. And by adding alkalinity to the ocean, it would essentially increase the ocean's CO2 uptake capacity. And the chemistry is fairly straightforward with this. So what's occurring now, as we dump CO2 into the atmosphere, we're actually acidifying the ocean. So as we add CO2 into the atmosphere, a large portion of that is absorbed by the ocean. And we have here the carbonate chemistry equilibrium equation. And so as we put CO2 into the ocean, it actually drives this equation to the right, acidifying the ocean with adding hydrogen ions. And so when we add alkalinity to the ocean, the hydrogen ions would be consumed by the dissolution products. And we would also be adding, in the case of, for example, limestone, we'd be adding calcium ions, which adds a positive charge. And then the combined effect would be to push this equation to the right and then allow the ocean to take up more CO2 in the process. So while the chemistry is fairly straightforward, what we don't know is, well, there's a lot we don't know, but one thing we're not quite sure about is how biology will respond. So that's where what our work has been focusing on for the past few years. And in particular, we look at phytoplankton in the Iglesias Rodriguez lab. And I have here just some pictures showing the diversity of phytoplankton you can find in the ocean. But essentially, phytoplankton are the foundation of ocean food webs. They're responsible for a large portion of the global net primary production. So through photosynthesis, they give us oxygen. They also draw down CO2 within the ocean and export carbon to depth. And some species also fix nitrogen and produce biominerals as well. And the two species we looked at are biomineral producers, Coccolithophore, which produces calcite or calcium carbonate, and then diatoms, which produce silica. So in our lab experiments, we set out to assess a representative of each of these, what we refer to as a functional group. So we chose Emiliania huxleyi, which is very common in the modern ocean and responsible for a large portion of calcite production. And we looked at a species of Coccolithophores, as Coccolithophores is the genus is one of the most globally abundant in the modern ocean. And we also set out to assess the biogeochemical implications of alkalinity addition. So how it affects nutrients, biominerals, and things like that. So we used two approaches, two alkalinity enhancement additions. We did one what we refer to as a moderate alkalinity enhancement, which we added 750 micromoles per kilogram of alkalinity. And then we did what we refer to as a high alkalinity addition, which we added 2,800 micromoles per kilogram of alkalinity. So that brought our total alkalinity within our cultures to 3,000 for the moderate and 5,000 for micromoles per kilogram for the high. And we selected these based on model predicted scenarios from the Renforth and Henderson 2017 paper. And to frame these additions, the natural variation within the surface ocean is typically between 2,150 to 2,450 micromoles per kilogram. And so we collected seawater from the Santa Barbara Channel, and we added both nutrients and alkalinity, the nutrients to ensure that our cultures were not limited by nutrients. And for the alkalinity additions, we use what we refer to as a limestone-inspired alkalinity addition. And we use this method because we found when using the actual, some of the proposed minerals, for example, quicklime, we couldn't get full dissolution and rapid and complete dissolution within our cultures. So using this method where we added sodium carbonate and calcium chloride dihydrate simulated the dissolution of limestone. And by doing so, it allowed us to bypass the dissolution kinetics of the minerals and just focus on the biological response to the chemical changes. So then we filtered out the biology, and then we aliquoted the water into our vessels. And we had triplicate vessels for each time point and for each treatment. And prior to inoculating our media with the phytoplankton, we bubbled it for about 3 days to bring the rest of the carbonate chemistry system, for example, the CO2, into the range of the model predicted scenarios. So what we found was encouraging for the additional research into ocean alkalinity enhancement's effects. We found that both Emiliania huxleyi and the species of Coccolithophores that we looked at both grew very well, even in very high alkalinity conditions. As you can see here, their cell abundances were well constrained, and there were no significant differences in their growth rates or their generation time. I won't go through all these in detail, but we also looked at organic matter production as well as biomineral production. So we looked at particulate organic carbon and particulate organic nitrogen and the ratio of the two. And we also assessed the production of inorganic carbon for Millionia huxleyi relative to organic carbon. So that this is referred to as the pick to POC ratio, which characterizes a calcifier as either on ecological timescales, whether it's a sink or a source of CO2. As kind of counterintuitively, calcification itself actually produces CO2. So conservatively, if the pick to POC ratio is below 1, the calcifier is looked at as a sink for CO2 on ecological timescales. And if it's above 1, they're characterized as a source. And so that's important in the context of ocean alkalinity enhancement, as if calcification were to increase relative to organic matter production or photosynthesis, it could actually add CO2 to the ocean, and then in the case of maybe like a bloom situation, potentially end up outgassing CO2 to the atmosphere rather than drawing down CO2. But what we found in our experiments were that organic matter production and the production of calcite was not statistically significant. There were no statistically significant differences in any of the treatment levels. However, what we did find at the bottom here, you can see, was an up to 3% reduction in the FE over FM, which is a proxy for the efficiency of photosynthesis. And so these values, I should note that these values are all considered healthy. They're above 0.6. The phytoplankton grew well. However, it's notable that we did see this moderate reduction of up to 3% in the highest alkalinity media. And we have some hypothesis for why that might have occurred, which I'll talk about brief here shortly. But first, I'll discuss what we saw for Coccolithophores as well. Again, here we looked at particulate organic carbon and particulate organic nitrogen, but instead of looking at calcite for the Coccolithophores, which don't produce calcite, we're looking at the silica. And again, we didn't find any statistically significant differences in the elemental ratios for Coccolithophores either. However, we did again see this slight reduction in FE over FM with the higher alkalinity media. And before I move on to why we think that might have occurred, I just want to note here, we also have some of our data points are noted with open triangles instead of the open circles. That's because we had some of our carbonate chemistry samples actually underwent reverse weathering while in storage. We don't think the alkalinity was affected while the Coccolithophores species were in culture, and we think that because essentially in collecting our carbonate chemistry samples, we filtered out during collection, we filtered them to remove the cells as well as mineral precipitates that we did see form while in culture. And these precipitates formed after that filtration. So we think that it's likely this occurred during storage, but we can't say so with certainty. But I mentioned that we did see some precipitates that did occur in culture, and we think this might be connected to the slight reduction we saw in the photosynthetic efficiency. That's because these precipitates that we saw had nutrients that are important to phytoplankton growth, such as iron, iron here, and phosphorus, and silicon, which diatoms need for their frustules. So this isn't quantitative, but this is something we're still assessing. But we, in addition to precipitates like this, we also saw a reduction in dissolved silicic acid, up to 18% in the highest alkalinity media. So we think that the reduction in micronutrients might have affected FE over FM values, but that's something we're still exploring in the lab. So just next steps and uncertainties that we drew from our experiments is kind of looking at why these reductions in the photosynthetic efficiency occurred at higher TAs, and what alkalinization's effect is on dissolved inorganic nutrients, as the nutrients we added are well above those that you would find in natural seawater. So whether similar reductions occur with natural seawater is something we're exploring. Also, what are the TA and pH thresholds to prevent reverse weathering, as we saw that reverse weathering in our carbonate chemistry samples. Also how to efficiently alkalize seawater, as we had to use the salts to mimic a ocean alkalinity enhancement scenario. And this is something that the community is exploring now. A lot of innovation occurring with methods to alkalize seawater, and also how phytoplankton community composition is affected. So we looked at single species, which is important in understanding the potential effects of OAE. However, you have to be cautious in extrapolating species-specific responses to ecosystems. So the experiments we're currently running, we're looking at phytoplankton community, how it affects the actual phytoplankton community as a whole. And with that, I'll turn it over to Deborah.
Thank you, James. And thank you, Will, for inviting us for the opportunity to present our results. So what I'm going to do for the next 10 minutes or so is going to, first of all, go back to my roots, which is on carbon acquisition by phytoplankton. And I'm going to go back to first principle questions or ideas. Then I'm going to talk very briefly about how we measure functional diversity. And then I'm going to finish with some ethical considerations, talking a little bit about ecological security. So I very much hope that there are some representatives of humanities and in the audience. So I will start with this slide about carbon concentrating mechanisms. And what you can see here is this really highlights the complexity of carbon acquisition. So we have all these dissolved inorganic carbon species in seawater here, and then we have membranes in different organisms. So we have been focusing on and this refers to the calcifiers on Coccolithophores, but this applies really to any calcifier, to corals, to foraminifera, and so on. So we have these carbon species crossing membranes in both directions, and there is one dissolved inorganic carbon species which is which does not go through biological membranes, although there are a couple of studies that might suggest otherwise. And then we have these two dissolved inorganic carbon species, CO2 and bicarbonate ions. And there are enzymes that catalyze the conversion of one into the other. For example, carbonic anhydrase. There are pumps. There are anti-port and syn-port mechanisms that control the fluxes of these carbon molecules going in and out. And the vast majority of photosynthetic organisms in the ocean have developed over time, over evolutionary time scales, carbon concentrating mechanisms to be able to take up bicarbonate ions as the affinity of Rubisco is very different from the concentrations of CO2 that they encounter in seawater. And so the vast majority of photosynthetic organisms in marine systems have these carbon concentrating mechanisms. And the reason why we chose diatoms and Coccolithophores, James, could you go down to the next slide, please? Is because they have distinct carbon acquisition mechanisms. So broadly speaking, Coccolithophores have relatively poor carbon concentrating mechanisms compared to diatoms that have more efficient ways of bringing carbon into the cell. So I chose these two graphs from, in my opinion, two seminal papers. We use these graphs in our lectures to tell the students about how different organisms respond to alterations in the different dissolved inorganic carbon species in seawater. So on the left, we have a graph from John Reinfelder paper in 2011. And on the y-axis, we have the Rubisco carboxylation to oxygenation activity ratio as a function of CO2 that you can see on the x-axis. The measurements were taken under dissolved oxygen concentrations of 250 micromoles per kilogram of seawater. And what you can see here is that, sorry, and I forgot to mention that the dashed line, horizontal line that you can see here corresponds to the carboxylation to oxygenation ratio of 2.5, at which 50% of photosynthetic energy is lost to photorespiration. And you can see that diatoms and coccolithophores are, for the most part, above this line. And in the case of dinoflagellates, which are another functional group, entirely different, it sits on the twilight between the animal and the plant kingdom, and it's functionally incredibly complex and different. Even at concentrations of 30 micromoles per kilogram of seawater, the carboxylation to oxygenation ratio of dinoflagellate rubisco is below the 50% energy loss level. So very distinct ways of acquiring carbon in these organisms. And then on the right, we have this figure from Ulf Ribosel's paper in 2004. And here we have the relative photosynthetic activity in percentage. And here on the x-axis, we have CO2. So we use this term in physiological studies to learn about the affinity of organisms for CO2 in relation to CO2 levels and their response. So this represents this is the half saturation constant, which essentially means the CO2 levels that achieve half of the maximum photosynthetic activity. And here what I'm showing is 3 representatives of diatoms represented in gray, skeleton imacostatum, and then we have 2 primnesiophytes, a non-calcifying one represented in black, which is called Phaeocystis globosa, and then we have a calcifying representative here, Emiliania haxillii. And what you can see here is that this half saturation constant is very different in non-calcifying phytoplankton versus calcifying phytoplankton. And there is huge diversity even within the calcifying groups. And within the, in this case, we have a silicifying phytoplanktonic organism and so 2 biomineral producers. But even within these groups, there is significant diversity. So the reason why I'm bringing this up is because we are paying attention to nutrients, which are key, super important potential impacts of metal toxicity, which is not brought up in this presentation, but it's extremely important. Many other factors, but the diversity in terms of carbon concentrating mechanisms within the phytoplanktonic community might be something that we need to pay attention to. And so far we are looking at in a way lab rats, organisms that we grow in the lab and we know very well. We can grow them very well as well. But now James is embarking, or he has been actually conducting several mesocosm experiments, and I'll tell you in a minute how we look at how organisms are doing within these populations and communities. Another thing I wanted to mention, 2 more things, is that from paleo-oceanographic studies, the aqueous CO2 in the tropical oceans during the glacial PCO2 minima, which represents the most extreme low CO2 levels that phytoplankton have likely experienced in the Cenozoic, are associated with upregulation of carbon concentrating mechanisms. So then this is done with modeling and some lab testing. So this is very interesting. And then broadly, calcification, which by the way is considered by some as a potential carbon concentrating mechanism, although there are studies that prove or disprove, this, in my opinion, very elegant proposal may respond neutrally or positively to increasing bicarbonate ions, given that bicarbonate ions are the actual source of carbon for calcification. So there are many things to consider, and I'm excited to see the results from the. Let's move on to the next slide. I'm excited to see the results from our mesocosm experiment. So James has been focusing on looking at communities recently, and I just wanted to mention that functional diversity and taxonomic diversity is really important to determine the health of ecosystems. And so we are using this tool called DNA metabarcoding, which essentially looks at amplicon sequence variation, which are the bits of the DNA from hypervariable regions within highly conserved markers like the 18S small subunit of the ribosomal RNA gene or the 18S ribosomal DNA. And for bacteria, which we are also looking at, it would be the 16S ribosomal DNA. So we are looking at essentially we are looking at the relative abundance of these ASVs under different conditions. So what I'm showing here is on the left is a study by Liu and others in 2022, and they apply this to Antarctic ecosystems to discover new, previously unreported harmful algal bloom forming species. So what you can see here on the left is an example of the kind of data that you get. So you get the relative contribution of the different groups. And then here on the right, you have a heat map with the relative abundance of these ASVs for the different organisms. Although we talk about species, I have to say that there is error propagation with the different steps of these methods, but we get reproducible data and we get very nice snapshots of what the communities look like under particular manipulation. So one of my former students, Dylan Catlett, in 2020 optimized this tool for a study that we conducted, a time series in the Santa Barbara Channel, and he keeps publishing results. There's another nice paper in which he compares that with pigment data. And then one of my former students as well, Tanika Ladd, we have a paper in review looking at the effect of ash deposition on the productivity. So it's, she used a similar approach, and it's a very nice way of looking at relative abundance in the control versus ash additions in this case. So essentially, James is doing the same with alkalinity additions to the system. Next slide, please. And in order to promote some discussion and something that I am particularly interested in is the environmental ethics, surrounding the work that we are doing and that we are proposing. And essentially, we are planning to do interventions that will have an impact on planetary scales, and we are altering biotic as well as abiotic processes within ecosystems, and this will undoubtedly change the social and policy setting. So it's whatever we do now is going to determine, in a way, the way the oceans will look for quite some time. And particularly interesting, in my opinion, are the feedback loops that either maintain the steady state or precipitate a rapid change from one state to another. Interestingly, the ethical considerations, in my opinion, are typically applied to the process rather than the outcomes. And the most interesting question is what outcomes are acceptable. So to determine to what extent ocean alkalinity enhancement can be pursued to promote ecological security in the context of resilience of ecosystems. So ecological security, how is this defined? So this is the commitment to the resilience of ecosystems themselves. So what does that mean? Before I move to my last slide, I have here the structure and domain of ecological ethics. So this involves biological research, conservation practices. There are professional codes of conduct established by bodies like the Ecological Society of America, Society of Conservation Biology, that feed into the ecological ethics of what we are studying. And then we have animal-specific ethics and environmental specific, and so on and so forth. So let's move on, and I'm keeping the ecological security this definition at the top. So the big question is, what criteria and principles do we use to determine what constitutes an acceptable outcome? So, for example, in the case of James' study, he found, for the most part, no responses. So his results are very much here in this scenario, in this outcome. And then we have coping and we have adaptation. And to me, it is unclear which is the most desirable outcome in order to maintain ecosystem or to promote ecosystem resilience. Ecosystem functionality is way too complex. So we need principles that can be context specific when needed, to determine, and we essentially need to determine what constitutes an acceptable outcome. So I love to hear your thoughts or any input into this, and I look forward to the discussion. Thank you all, and I look forward to the questions. And before we leave, I would like to thank the associated students, Coastal Fund, University of California, Santa Barbara, the Carbon to Sea Initiative, the Climate Works Foundation, and the Pat Tillman Foundation that funded the work that we are presenting today. And I'd like to thank our colleagues from the University of California, Santa Barbara, and the Natural History Museum. Thank you all.
Thanks so much, James and Deborah. So yes, now we'll move to the question phase, and I'll just start with a couple. And as I said, encourage people to start populating the Q&A, and we'll proceed. So one thing that you suggested, Deborah, was the issue of metal toxicity, right? In the context of ocean alkalinity enhancement and some of the other approaches that we look at. And it's my understanding in this study that you were focused on limestone interventions. Do you think that the results in terms of phytoplankton functional groups or other species might be different if the interventions involved minerals such as olivine, for example?
Sorry. Yes, undoubtedly the effects will be different. Now, when we look at short-term responses, our experiments are very much abrupt manipulations. So you look at just a few generations, they might not show any impact in terms of toxicity. And in fact, there are a few studies on olivine specifically, like by Dave Hutchins' group at the University of Southern California. And there are a few studies, and also by the group in Guiomar in Germany, I believe. And we are not focusing on that because olivine has, cadmium, nickel, chromium residues. But I think it's important to think about the consequences beyond just a few generations, because organisms undergo acclimation, but also longer-term adaptative processes. So, these results are important, and they are very interesting. I also think that we need to think about, how communities respond in longer time frames. And of course, it's very difficult to test experimentally because these experiments have important biases. So in fact, I try to stay away from those experiments because they are difficult to run and to interpret the results. But yes, the results are alkalinity specific for sure.
Yeah, that leads me to a branching question from that. As you indicated, that this was a short-term experiment, right? It was a snapshot, and so it seems like you expressed some trepidation in terms of if conducted on a longer scale. But obviously, some of these impacts, well, maybe not obviously, but perhaps some of these impacts could be different in the long term if there's aspects of biomagnification or other considerations. Do we need to be confident from an ethical perspective, from a scientific perspective, about the impacts of ocean alkalinity enhancement? Do we need to run longer experiments? And if yes, what kind of timeframes are we looking at? How much does it increase funding requirements, things of that nature?
Yeah, I mean, long-term experiments are tricky, and there are important biases to consider because, for the most part, you use clones of organisms. You use a bunch of representatives of, or even sometimes one. So they are pretty artificial systems. I mean, when I think about manipulations, and it is very tricky to do, but when I think about manipulations, you know, and organisms mutate, you know, and evolve. So there are, if you like, tempo of evolution, or, you know, mutation rates or evolutionary rates associated with specific types of organisms. And then you have, you know, these large temporal scales of change on Earth that one can revisit and say, okay, how can we experimentally reproduce that? It's very tricky, you know, to do that. And for me, the trickiest part is to, you know, how do you interpret the results when organisms are, you know, in a way fed in isolation? Even if you mimic natural responses, you know, it's very, you know, for one thing, the vast majority of the bugs that we study undergo sexual reproduction, which, you know, leads to higher genetic variability. And, you know, in culture, we have no idea. There might be, when you grow several strains together, you know, we have seen that in dinoflagellates, but, you know, for the most part, you grow the organism in very artificial conditions. So I try to stay away from that. So I think a combination of modeling and find a way to, you know, to mimic what Earth has seen. You know, in fact, a big change is probably undesirable. And another thing to consider is that for our experiments, we have paid attention, and I think most experiments out there, you know, are paying attention to the natural variability, and we have tried very hard to stay within natural variability, so we don't go off, you know, too much. So yeah, I don't know if I've answered your question, but yeah, very, very difficult experimentally, things to run.
Yeah, I think that's it's kind of a fascinating conclusion, right? Because in some ways, intuitively, we know that we would like to be able to assess on a longer term, given the fact that that the snapshot may not give us the picture in the future, but it sounds like it's very difficult and has its own bedeviling factors to conduct the longer term experiments. So it's interesting how to decide as a policymaker, for example, how to proceed under those kind of conditions of uncertainty, right? That seem a bit intrinsic. So.
I should say, I just add very quickly that, you know, natural analogs can be very informative. If we look at the environment where, you know, alkalinization, typically close to terrestrial systems, has taken place, I think we can learn a lot more than running, you know, lab experiments in that context, in the context of adaptation.
Yeah. Are we doing a lot of that research? Do we need to do more of it?
I think we probably need to do more of it. I mean, the natural analogs, most of them are rather imperfect, but so are the paleo data, you know, that we are using it, but it can be extremely useful. I mean, in the ocean acidification, you know, world, we have looked at the past to infer, you know, basically to learn from, yeah, rather imperfect analogs for ocean acidification. Like, we looked at the Paleocene, Eocene thermal maximum, and, you know, looked at any changes in diversity of you know, calcifiers, for example. Groups have looked at that. So I think I would like to see more of that. Yeah.
Okay. Anything to add in this context, James?
I would just say, you know, I agree with Deborah with, you know, needing more data to essentially assess, you know, looking at environmental effects. And yeah, I think lab experiments tell us a lot, but you know, I think the best thing as far as continuing to get more data and helping us make decisions as a society would be, you know, these small scale field experiments that are, you know, carried out on extended timeframes. I think, you know, moving from the lab to the field is always, it adds a lot more complexity. You have confounding factors that, you know, it's hard to disentangle maybe alkalinity's effect based, you know, on, you have grazing and perhaps effects on nutrients. So disentangling the complexity there is more difficult. But I think that's going to give us the best, you know, snapshot of really understanding what might occur in a real world deployment. And I think that should really kind of be the focus. So I think I would just add that on to kind of tack that on to what Deborah had mentioned.
Okay. All right. So in the study, obviously you focused on representatives of a couple of PFGs. Do you think there's other important phytoplankton functional groups that we should be looking at in the future in terms of potential impacts of OAE?
Yeah, I would say there's quite a few, and like you have nitrogen fixers. That would be an important group to look at. Also, diet, like your harmful algal bloom species are very important to look at. If alkalinity enhancement for some reason promoted their growth, that could have some large impacts on ecosystem. So I think really there's many of them, but I think probably the best way of trying to assess that is these field experiments and using methods like DNA analysis and stuff to look at shifts in community composition. I think that's probably the best way of trying to parse that out. So.
All right, well, that's a natural transition to our first audience question, which focuses a bit on contours of field experiments. And the question is, any alkalinity added to the ocean will be quickly diluted, so the bioexposure to high alkalinity will be brief, in contrast to beaker experiments and mesocosms. How will this dilution over time be experimentally addressed to more realistically represent what happens in OAE?
So colleagues are already moving into the field slowly, running deck incubation experiments and using tracers to look at the impacts of alkalinization and how can they be traced. The big issue is MRV right now. Of course, is monitoring, reporting, and verification. But I think the community is working very closely together to really tackle that through both experimentation as well as modeling. But yeah, I mean, in my opinion, we need to move to the field to run experiments, to make observations, to conduct trials, and it will take some time to get it right, but we really need to move to the field. So getting permits should be a priority. It is a priority.
Okay. All right. So you brought up ethics, and here's a question that allows us to tease that out on an even more sort of macro perspective. So the question is, mining of any rock is highly destructive. Does OAE LCA life cycle assessment include those negative outcomes on land that still make the process ecologically, quote unquote, sane?
That's a really good question. Do you want to tackle that, James, or do you want me to?
I mean, yeah, I think it's a huge ethics question, and I don't think there's quite an easy answer to that at this point. And that's something that's going to have to be weighed out, the impacts of these mining operations, and also outside of just the ethics behind the mining itself, the energy that it would take to grind down these minerals and transport them is another consideration. So I think it's something that the community is looking at and investigating, but I don't think there's an easy answer to that at this moment. I know to add on, there's also the innovation that's occurring right now. There may not be a need to mine minerals out in high quantities. For example, there's a lot of innovation going on for electrochemical methods where alkalinity is produced from seawater itself, and different technologies that are being explored there. So I don't think it's I guess I don't really have a great answer for that. I don't think it's an easy question, but it's some that's definitely a huge ethics issue behind ocean alkalinity enhancement. I don't know if you have anything to add on to that, Deborah.
Yeah, I've been thinking about that, of course, a lot for the last year and a half or so, and I have to declare my bias to electrochemical methods. I really yeah, I think, on several fronts, but I think the environmental impact of mining, transportation, industrial processing, etc., is too great. And then, for those of you who read the National Academy's report, we have this cartoon that highlights, the problems of adding particles, that do not dissolve readily in the surface ocean. And we did try several minerals, and we only got partial dissolution at best, and the dissolution rates, the dissolution kinetics, it was just too, experimentally was not viable. And of course, if you plan deployments at sea, it would be a very undesirable method. We need engineers to work it out. But if you think about the cost, the environmental cost and the economic cost, in my opinion, and this is my bias, and in my opinion, but I think the effort should be on electrochemical methods rather than on mining rock to pulverize and add to the oceans.
Do you know of folks that are involved in kind of parallel assessments of ecosystem impacts of the electrochemical approaches at this point?
Well, Phil Ramforth and other colleagues at Harriet Watts are looking at, have been looking at life cycle assessments. Mm-hmm. And I believe that includes liquid, if you like, liquid alkalinity, sodium hydroxide, the electrochemical, method behind. Everything comes at a cost, you produce acid, you have to, it's not exactly clean, but with a smart way of, using the byproduct, if you like, I think it would be better environmentally than the alternative, than using rock. Sodium hydroxide is clean. We don't have any residues in the mineral that could have long-term effects if deployed regularly. So I think, in my opinion, it's a better approach in terms of ecosystem impacts and long-term biogeochemical impacts as well.
Okay. Next question. This is for James. You pre-filtered your water after TA and nutrient application. Could that have contributed to precipitation in the high TA because of possible precipitation nuclei in your unfiltered water?
Well, the last portion there, the unfiltered water.
Yeah, that's what it said.
So all of our water was filtered, prior to actually aliquoting that out into the media. I think that we filtered through a 0.2 micron mesh after adding the alkalinity and nutrients. So any precipitates that formed previously, I imagine would be drawn out at that point. So I don't think so. I don't think that there should have been nuclei that could have pulled stuff in at that point. And we think it was likely more based on the nutrients that the dissolved nutrients that we added, that maybe formation of iron oxides, for example, with changes in pH that then leached minerals out. That's kind of what our hypothesis is of what occurred there. I don't know if I explained that in the best way there. I don't know if I answered. But I don't think so. No, I don't think there would have been precipitates post filtration. Right.
This is a question related to the small decline in Rubisco function at elevated alkalinity. Is anyone exploring potential interactions of this mechanism with directed evolution work at Scripps, Ahmed Badrian's lab, to radically accelerate photosynthesis?
I don't think so, and that would be really, really interesting. Yeah, I don't think anyone is looking at that. I mean, this is it's an open field. You know, this is really an emerging field. It's something that I'm really interested in. Is, you know, we tend to look at inorganic compounds, and you know, because they are the focus of attention. But you know how this affects organic chemistry as well. You know, it's fascinating, and we are, you know, it will take some time until we get there, I think. But no, I don't think anyone is looking at this. Have you come across any papers, James, to do with this? I haven't.
Yeah.
Or I don't think anyone is looking into this. Very interesting.
Okay. This is a question that's related to my broader question about looking at other critters. It says, what kind of conclusions can be realistically drawn to other forms of sea life? Are these species particularly sensitive, and you can assume other species are resilient as well?
No, we can't assume that. Absolutely not. We started with these two representatives because they are really important ecologically, because we can grow them really well in the lab, because we know a lot about them from the literature. But we absolutely have to look at, you know, for example, dinoflagellates and diatoms. Bacteria are really important in the system as well, and we are beginning to look at them. So no, the answer is no. And I don't think these two species are particularly sensitive compared to others. And we, you know, the community has focused in the community working on carbon acquisition in photosynthesizers, in phytoplankton, has focused on ocean, you know, acidification rather than alkalinization. But yeah, I don't think these two species are particularly sensitive. Yeah. But yeah, we cannot extrapolate our results to any other functional groups.
Anything to add there, James?
Yeah, and I would just say, even within you know, within the functional groups themselves, or within just diatoms themselves, there's, if you in the literature, for example, with ocean acidification, the species specific response can vary quite a bit, even, you know, within a functional group. So yeah, definitely can't be extrapolated out to other functional groups, or even within you know, you can't be applied to all diatoms either, for example.
Mm-hmm. Let's see. Quick logistics question here, which is, will recording of the talk be available afterwards? The answer is yes. Usually we post in about 2 to 3 days on our website. Okay, next question. I think you measured this, but it asks, what was the CDR achieved in your experiments?
So the uptake, CO2 uptake capacity increase was 80% higher. So when we added the, like for the 5,000 micromoles per kilogram, it increased the uptake CO2 uptake capacity by 80%. And for the moderate, it was around 20 to 25%, I believe.
Okay. Is there any preliminary evidence to constrain the potential contribution of optical change, more turbidity from OAE to the change in photosynthesis at elevated alkalinity?
Well, I mean, if this is quite possibly, yeah, quite likely, in fact. I mean, what we did was limestone inspired. We call it limestone inspired because we mimic the chemistry, because we got partial dissolution. So we didn't have to deal with any, you know, particle effect on light attenuation. But I'm sure if I mean, and this solution is very unlikely to occur rapidly if deployed in the ocean. So I'm sure that will have an effect on light irradiance, and there will be light attenuation with the release of the particles, but I'm sure that will be looked at because that's one of the key measurements that are taken at sea.
Let's see. This don't think this was really part of the study, but if you have opinions on this, there's a lot of uncertainty in MRV of diatom-based carbon sinks. What's your perspective on this?
Yeah, I mean, the problem is that the signal to, I mean, when one envisage what going to the field is going to look like, the signal to noise ratio is very low. It's going to be very low, so it's very difficult to actually produce any data with the tools that we have right now with any degree of certainty. But yeah, the community is working, very hard to, to come up with a model to begin to tackle these uncertainties. And David Ho and Katia Fennell and many others are behind this kind of work. But yeah, I mean, that's the big worry. It's like, how do we, when we go to the field, we need to provide some numbers, and we need to test if it works, and it's yeah, that's the weakest aspect of the whole ocean CDR, oceanity enhancement, planning for theoretical planning for going to sea. Yeah.
Anything to add in that context, James? MRV?
No, I don't think so.
Yeah. I guess one last question is again, you raised ethics toward the end of your presentation and questions of security. How do scientists then in this ecosystem where they're going to be providing some of the data that could result in large scale interventions in the world's oceans, right, that could have long-term implications, right, both positive and negative, what do you view your role in the interaction with stakeholders or with society writ large in terms of addressing these issues, talking about trade-offs, helping people to formulate decisions in this context from an ethics standpoint?
Yeah, I think science has to inform. I often think about the honest broker position, so the scientist, even if, the scientists might have, their opinion on the way for, we need to provide information. We need to operate in a more transparent way. We academics are terrible for the most part because we release our results when the paper, is out there, and we cannot really afford to do that in this context. I mean, I think it's important to protect your intellectual property, but at the same time, we have to come up with mechanisms to release the information so that the field can move forward fast because there is an urgency, while at the same time being protected. Yeah. And I think the role of the scientists is, I think, for example, the activities by the private sector and by policy makers have to be guided by scientists, and we need individuals like you who can talk to scientists and who can talk to environmental lawyers and social scientists, and I think even if we have our niche and our we sit in our box and we do our work, I think more and more we will need to to work together with people who understand the rights of tribes, and So I think that's probably going to change.
Otherwise, I'll remind you in an hour.
Yeah, that's it's a weighty responsibility on all of us at this point, especially when we hear a lot about how quickly we need to move, right, given the climate crisis, and yet how we proceed with uncertainty. So I appreciate those insights, and more generally, just really appreciate James and Deborah, you coming on today and summarizing this important research, kind of helping us frame what we can learn from it, what more needs to be done in this field, and really appreciate it. As always, appreciate all of the audience questions. I hope that many of you will join us again next month. We'll have a couple more in this series. And until then, thank you, everybody, and goodbye now.
Thank you. Bye-bye.
Thank you.